In last video, I tell you what then would be the phenotype of a plant that had a genotype capital T small t, because it is asking about the phenotype, and we know phenotype means what? Physical appearance, right? So physical appearance is what is the physical appearance of this plant in which genotype is capital T small t. We understand that because capital T is present and capital T is a dominant character or dominant trait. So this plant is tall. What is the phenotype? It is tall, right? And what is the genotype? It is capital T small t, right? Okay, today we start from that portion as Mendel found, because all the lines which is given here, all the lines means these lines, they come in statement type questions or accessory type of questions, multi-statement type. Sometimes they can question come from this segment, right? So we have to read all these statements line by line. As Mendel found, right? It is very broadly explained, right? You get bored when we read this one, you get bored. But one thing is there, we have to read, right? Very simple explanation is given okay, I will tell you after this explanation what is given in this chapter. It is in a single unit. It is how a Mendel explained it statically or mathematically, that is explained here. And he used this simple formula A plus B square is equal to, right? That A plus B square means what? A square plus 2AB plus B square. That is given simply here, right? The capital T means what? That is in F2 generation, we know we get 1:2:1 ratio. 1 means what? Capital T, capital T. Capital T means what? It is T square. Then capital T small t, 2 times we get it here. So capital T small t, 2 capital T small t is there. And small t means what? T square, small t square is there. That's this mathematical or statical, this equation, this means we can say this equation, right? It is also used by Hardy-Weinberg in case of population, right? In case of population, Hardy-Weinberg, P square plus Q square, P square plus 2Q, PQ plus Q square is equal to 1, Hardy-Weinberg equation in case of population. In another chapter, we will read this one. That is here. So we read this one line by line. First, as Mendel found the phenotype of F1 heterozygote capital T small t to be exactly like the capital T parent in appearance, means exactly like the tall plant, right? In appearance, he proposed that in a pair of dissimilar factor, then he understand only one can express and another cannot express. Then he get one dominates the other, as in F1 generation. Through F1 generation, we get the law of dominance, and hence it is called the dominant factor. Which one is dominant factor in capital T small t? This capital T is dominant factor. So dominant factor is always represented by capital letters, not T, but capital letters, right? While the other factor is recessive. Recessive means which cannot express itself in F1 generation. In this case, capital T for tallness is dominant over small t for dwarfness. That means tallness is dominant over dwarfness, right? That is recessive. Dwarfness means recessive. He observed identical behavior for all the other traits were in his studies, because he took 7 traits, 7 alleles, or we can say 7 contrasting traits by using of 14 pure breeding plants, right? He get this law of dominance in all the cases. One is dominant on another. He simply observed that in F1 generation, when we cross between 2 opposite or contrast traits, what happens in F1 generation? Only one expressed, another one is not expressed. Which expressed, this trait is called dominant trait, and which is not expressed, this one is called recessive trait. And this means law is called law of dominance. This is the first law of inheritance, Mendel's first law. It is convenient and logical to use the capital and lower case of an alphabetical symbol to remember this concept of dominance and recessiveness. Now here it is given here why we use capital or small letters, right? That is one. Do not use capital T for tall and D for dwarf. Why? Because you will find it difficulty. We will find it difficulty to remember whether T and D are alleles of the same gene, same characters, right? Okay, in one case, if we select only one trait, then we can understand. But if more than one traits are there, so many traits are there, then we get confused, right? Then we get confused. D means it is dwarf or D means it is another, right? But when we start capital T, small t, so we understand capital T means only capital letter means it is dominant, that is tall, and small t means not a dominant. It is just opposite of tall, right? So here, T and alleles of the same gene or character or not. Alleles can be similar as in the case of homozygote capital T and small t, or can be dissimilar as in case of heterozygote. Heterozygote means what? Capital T, small t. Since the capital T, small t plant is heterozygous for gene controlling one character, that one character is height of plant, length of plant, right? It is a monohybrid, and because only one trait, here we use only one trait, tallness or height of plant, so it is crossed. This one is called monohybrid cross between capital T pure breed and small t pure breed, means a long pure breed and a dwarf pure breed, and it is called as monohybrid cross. Mono means one, hybrid means contrasting traits are, right? If we use two traits, then it is called dihybrid cross. If we use three traits, then it is called trihybrid cross. 4 tetrahybrid, 5 pentahybrid, so on, means polyhybrid cross. Okay. From the observation that the recessive parental trait is expressed without any blending in F2 generation. In F2 generation, already yesterday we discussed this one, and I tell you that in F2 generation, what we get? In F2 generation, we get one pure capital T, two hybrid capital T, small t, and one pure dwarf small t. No blending is there, without blending. That's why in F2 generation, or we can say F2 generation, prove the law of purity of gametes. First thing is there, because there is no blending, they segregate purely in their pure form. They segregate small t, capital T. This is pure breeding line. This one is also pure breeding line of dwarfness, and there is no blending, right? So it is called as law of segregation. First thing is there, because here these two gametes, we can say these two genes, they get separate freely, right? Second thing, no blending is there, they separate in their pure form, so it is also called as law of purity of gametes. Okay. In F2 generation, we can infer that when the tall and the dwarf plant produce gametes by the process of meiosis. Now here, how they segregate, right? At that time, at the Mendel time, he did not get this idea, but this time we know gametes, they are formed through the meiosis, right? Because these plants are sporophyte plants. Sporophyte means they are diploid plants, and in all the sporophytes, right? All the sporophytes, because they are diploid plants, so gametes are formed through meiosis, right? Okay, when you see the bryophytes, because bryophytes mean they are already haploid structures. So what happens? Their gametes are formed through mitosis, right? Okay, because meiosis occurs in very early, right? In the case of after the formation of spores. So all the sporophyte, because this plant is also sporophyte, so here what happens? Gametes are formed through meiosis, right? By the process of meiosis, the alleles of the parental pair separates or segregate from each other, and only one allele is transmitted to a gamete. When the chromosome they get separate, and in which phase chromosome gets separate? You know, in which phase chromosome gets separate? We know anaphase first, in meiosis first. Anaphase first, in meiosis first. That is the phase. Which step is anaphase first? At that time, homologous chromosomes they get separate. Anaphase second is not correct, because anaphase second, what happens? Anaphase second, chromatin threads they get separate. But in anaphase first, homologous chromosome they get separate, right? As for example, if it is a human, you know, human means it male human. When we consider the male human, so we know here two types of gametes are formed. In male human, two types of gametes are formed. Which one? One is in which X chromosome is present, another in which Y chromosome is present. 50% chances is there, right? X and Y they get separate in anaphase first. At that time, X chromosome, they are homologous chromosome. 23rd pair means one homologous chromosome. So this 23rd pair chromosome, it gets separate. When gets separate means what? One cell, one daughter cell in after anaphase, what happens? One daughter cell formed. One daughter cell means one gamete. We can say here haploid number of chromosomes are there. You can see here. This when they get separate, this one is homologous chromosome 2, right? When they get separate, then what happens? Suppose that this one is Y chromosome, right? This one is X chromosome, this one is Y chromosome. When they get separate, then what happens? One side X chromosome is going on, these are all the chromosomes, and this side Y chromosome is going on. After then what happens? This cell, it divides into two parts, right? Through furrow. One gamete, it contains X from 22 plus X, and another gamete, it contains 22 plus Y. I think it is also explained somewhere else, right? So two types of gametes are formed. These two types, this means differentiation. That means gamete separation of chromosome occurs in anaphase first, right? That is given here. By the process of meiosis, the alleles of the parental pair separates or segregates from each other, and the only one allele is transmitted to a gamete, right? This segregation of alleles is a random process, and so there is 50% chances of a gamete containing either allele, as has been verified by the result of the crossing, right? In this way, the gametes of the T plants have the allele only T, because both are T means in anaphase first what happens? We get only T. Only one type of gamete is formed, 100% one type of gamete is formed. As for example, in female humans, only XX chromosome is there, 23rd chromosome is XX, 22 plus XX. Then how many types of gametes are formed? In a female, we know only one type of gamete is formed, it is 22 plus X. Another one is also 22 plus X, means both are same. But in case of males, what happens? One means 50% it is X, 22 plus X, and that 50% 22 plus Y. That is given here. Because T, both are T, means only one type of gamete is there, and it contains T. And the dwarf, which is small t, right? It also contains only one type of allele, that is small t, right? During the fertilization, the two alleles, T from one parent, say through the pollen. Similarly, if you think this is a pollen, means it is a tall plant which contains pollen, right? And small t from the other parent, that is female, through the egg. Female plant is small, dwarf plant is female, and tall plant is male, right? Are united to produce zygote. They fuse and they produce zygote that have one T and another one small t allele is there. T and small t both are because they fused. So both chromosomes, they come into a single zygote, right? So T, small t both are there. In other words, the hybrid have T, small t. This one. In other words, the hybrid have T, small t. That's why it is called hybrid, right? Since these hybrid contains allele which express contrasting traits, the plant are heterozygous. T, small t, both traits are present in the same zygote. This one is called heterozygous. Two different type of alleles are present. The production of gametes by the parent, the formation of zygote, the F1 and F2 plant can be understood from the diagram that is called Punnett square. Punnett square first, because this one, right? This is Punnett. Who is Punnett? Is even it was developed by a British geneticist, Reginald C. Punnett. That is the name of this scientist. It is a British genetist, Reginald C. Punnett. So it is called as Punnett square. One Punnett square is given here. This Punnett square, right? It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. Possible, we can understand. It is very easy, right? Okay, we will also is understood through dihybrid cross. Okay, the possible gametes are written on two sides, usually the top row and the left column. That two sides, right? All possible combinations are represented in the boxes below the square. This possible T, small t. You can see here this T, this small t. this capital T, this capital T, this capital T, capital T, capital T, small t. Now here small t, capital T and small t, that is given here. You understand, right? Which generates a square output form. Here you understand what is the phenotype ratio? We understand here. Phenotype means what? Look, capital T, 1, capital T, capital T, small t, these two, this 1 and this 1. Means 1:2:1. That is the genotype ratio. And phenotype means what? Because capital T is present, this one is tall, this one is tall, this one is tall, but this one is dwarf. Means 3 tall and 1 dwarf. Right? The Punnett square shows the parental tall, capital T, male, and dwarf, small t, female plant, and the gametes produced by them, and then F1, capital T, small t progeny. We get all the plants are tall. The F1 plants of genotype capital T, small t are self-pollinated. Means selfing. This one is called selfing. When self-pollinated, then what happens? The symbol, this is the female symbol, you know very well, and this is the male symbol, right? Are used to denote the female X and the male pollen of F1 generation, right? Respectively. The F1 plant of the genotype, right? Capital T, small t, when self-pollinated, means through selfing, produce gametes of the genotype capital T, small t in equal proportion. 50% genes means gametes are capital T, they contains capital T, and 50% gametes they contain small t, right? And when fertilization take place, the pollen grain of genotype capital T have 50% chances to pollinated X of the genotype capital T, as well as 50% of chances, right, of genotype small t. 50% chances is there. Also, pollen grains of genotype small t have a 50% chance of pollinating as X of genotype T, right, as well as genotype small t. Means possibility is 50% chances, right? There is no exact chances. As a result of random fertilization, the resultant zygotes can be of the genotype either it is capital T or it is capital T small t or it is small t, right? From the Punnett square, it is easily seen then when we consider the Punnett square, so then we understand, right, in F2 generation, in case of F2 generation, because F1 generation means 100% they are tall. But in case of F2 generation, what happens? 1/4 of the random fertilization leads to the formation of capital T, right? Capital T, 1/4, and half 50% means 1/2 lead to capital T small t hybrid, and 1/4 are pure breed, that is small t. Through the F1 have a genotype capital T small t, right? But the phenotype character seen in tall. Phenotype character in this one means it is a tall. You know, phenotype is hybrid, but phenotype is tall, right? And when we consider the F2 generation, at F2 generation, 3/4, because it contains capital T means it is tall, it is capital T small t, capital T is present and capital T is the dominant character. So phenotype is also tall, but this one is only small t, no dominant character is there, it is dwarf. We understand this 2 plus 1, 3, right? 3/4 of the plant are tall, whereas some of them are in which some are capital T, that is pure breed tall, and others are hybrid tall. Some. Externally, it is not possible to distinguish between the plant with the genotype capital T and capital T small t. Externally, right? Phenotypically, we cannot understand which one is hybrid and which one is, right, pure breed line. So, for this, we have to go in cross, that which is called test cross. Okay, we will explain after this one, right? Hence, within a genotype pair, capital T small t, only one character capital T is expressed. Hence, the character T or tall is said to be dominant over the other allele, that is small t or dwarf character which is called recessive trait, right? It is thus due to this dominance of one character over the other that all F1 are tall, because in F1 all the zygotes they contains capital T small t. So in F1 generation, because capital T is present in all the seeds, that in all the zygote, that's why all the plants they are tall, right? Though the genotype is capital T small t, and in F2 generation 3/4 of the plants are tall, though genotype is 1 to 2 are capital T small t, because capital T is present, dominant trait is present, so it is tall, and only 1/4 because both tall allele is present, capital T is present, dominant trait is present, so it is also tall. So 1/4 capital T, pure breed capital T, half hybrid capital T small t, and 1/4 it is small t only. This lead to a phenotype ratio of 3:4 tall, 1/4 tall, right? 1/4 capital T and half capital T small t, and 1/4 small t. That means we can say 3:1 ratio phenotype ratio, but genotype ratio is 1:2:1, right? Already we will read this one in last class, but right here it is. Now here, how mathematically he explained or statically he explained, right? Now here, the 1/4, 1:2 or 1/4 ratio of capital T small t, capital T small t is mathematically condensable to the form of binomial expression, that is ax plus by square whole square, that has the gametes bearing capital T or small t in equal frequency of half, right? That is 50%. The expression is expanded as the given below. What expression here? 1/2 t, 1/2 means half t plus half t square is equal to half t, right? That one multiply by half t and half, we can say here, and we get 1/4 capital T small t, capital T, 1 second capital T small t when you expand this one, right? And 1/4 it is small t. That is the mathematically, right? Simply here it is given here 1/4 capital T, 1/2 means what? And when you means if you means what happens here? If you divide it from 2, then what you got? You get t square plus 2 capital T small t plus small t square. This is the same, this one, right? 1/4, 1/2 and 1/4. Simply you can see here 1/4 means what? If I get square means I get here 1/4 square means 1/2, 1/2 square means 1 and 1. It is also simply we can say, right, this one. Mendel self-pollinated the F2 plants and found the dwarf F2 plants continue to generate dwarf plants in F3 or F4 generation. This one capital T and small t, right? He found that they continued to generate dwarf plant. This one dwarf plant, and this was only long plant, tall plants in F3 and F4 generation. He concluded that the genotype of dwarfs were homozygous. You understand? That means after self-pollinated, right, of this one, in selfing, after selfing he get continuously small t. In every generation he gets small t, means dwarf plant only is there. So he understand that means here only homozygous condition is there. In this case, means tall plant means he cannot understand, because phenotypically we cannot understand which one is pure breed or which one is hybrid, right? But dwarf plant means we understand when it is dwarf. Phenotypically we understand this one is dwarf. That means it is homozygous. What do you think he would have got had the self-pollinated of tall F2 plant? So when he self-pollinated the F2 plant, some plant which contains capital T, they continues gives capital T, capital T, or capital T in F3 generation, F4 generation, or F5 generation. Every time he get only capital T, capital T, capital T, capital T, capital T, capital T, because it contains pure wheat. That means he understand this specific is pure wheat. But when he use this one, capital T smart, right? Now he get once more, means capital T, capital T, capital T smart T, capital T smart T, and smart T smart. Simply, no, this one is pure, this one is pure. Now he, another means what happens? He get capital T, 2 capital T smart T, and smart T smart T. Simply this one is separated. The law of segregation happens in every generation. This one is F3 generation, this one is F4 generation, and so on. Hybrid seeds or hybrid gametes, they segregate. But pure breed means there is no segregation. Small T. Pure capital T means some seed they are not segregated. We can say here in F2 generation, in case of this one, he asked what would he got have his self what do you think he would have got had he self pollinated a tall F2 plant? Then he got some seeds they continuously give tall plant, but some seeds they segregate into 3:1 phenotype, right? So seeds which segregate into 3:1 phenotype, they are hybrid, and which is not segregated, which gives every time only tall plant is there, that means they are pure breed, right? From the preceding paragraph, it is clear that though the genotypic ratio can be calculated using mathematically probability like this one, right? By simply looking at the phenotype of a dominant trait, it is not possible to know the genotype composition. That is, for example, whether a tall plant from F1 or F2 has capital T small t, or capital T. We cannot get, right? Composition cannot be predicted. Therefore, to determine the genotype of a tall plant, right, at F2 generation, Mendel crossed the tall plant from F2 with a dot plant, and that is called he called a test cross. Right? So test cross means what? Test cross means the offspring is crossed with one recessive parent. This one is important, right? Recessive parent, right? Back cross means what? Back cross means, okay, test cross is a type of back cross. Why it is called back cross, right? Look here, back cross means two types. One, when it is crossed with the tall plant, means dominant parents. When it is crossed by the dominant parent, then we have no idea, means all the parents, all the offspring, we get only dominant, means we get only tall, means dominant traits are there. Phenotypically, we cannot understand which one is hybrid or which one is pure or which one is, right? But when we use a recessive parent, means when we cross a recessive parent with a offsprings, right, then we get the phenotype and genotype both are same ratio, right? Phenotype genotype ratio is same when we cross with recessive parent. So by seeing the phenotype, we can get the idea which one is pure breed, which one is hybrid, right? So it is called as test cross. In a typical test cross, an organism for here pea plant, right, showing a dominant phenotype and whose genotype is to be determined, each cross with the recessive parent instead of self crossing, right? Recessive parent, self cross, right? This, and that one is called test cross. The progeny of such a cross can easily be analyzed to predict the genotype of the test organisms, because genotype and phenotype in a test cross, genotype and phenotype both are same, right? Show the results of a typical test cross where violet color flower is dominant over white color flower. Using Punnet's square, try to find out the nature of offspring of a test cross. What ratio do you get? First thing is given, and using the genotypes of this cross, can you give the general definition for test cross? Definition for a figure is given, we will read this one. A test cross, right? Okay. First, because we are going to read the test cross, First I explain you here. How a test cross helps to determine whether the plant is hybrid or a pure breed line. Test cross. In test cross, what happens? Look here. Here, offspring is crossed with recessive parent. That is called test cross, right? And it is a type of back cross. This one is a type of back cross. How is it explained? Suppose that we have three types of seeds. One is capital T, another one is our capital T, small t, capital T, small t, and third one is our small t, small t. Right? These three seeds are. How could we understand which one is hybrid or which one is pure dwarf or which one is pure tall? Right? Suppose these are offsprings. This one is F2 generation. F2 generation. In F2 generation, we get capital T, small t, and small t, small t. Right? When we cross this first seed with recessive parent, so recessive parent means what? It is capital T, small t. Right? So here, capital T, we made a square. First, we should make a square. Right? So which gametes are present here? We understood because only capital T is there. Only one type of gamete is there. It is capital T. This side offspring genotype is there, and that side it is parents. Right? Recessive parent. Recessive parent means it is small t. Now in this first case, what happens? We get capital T, small t, capital T, small t, capital T, small t, and capital T, small t. Means we can understand here because in all the case, capital T is present, dominant trait is present. So all the plants, they are tall plants. 100% tall plants. Right? Phenotype 100% tall plant. Right? And 100% tall plant means what? This means we can understand that offspring is pure T, tall. Right? This is the phenotype. 100% tall. And genotype means we understand the offspring genotype is capital T, small t. This, right? Okay, now in second case, when it is crossed, when a recessive parent is crossed with a hybrid offspring, then what we get here? This side it is parent, means small t, recessive parent, and that side because it has capital T, small t, means 50% gametes are capital and 50% are small. Probability is 50-50%, right? Here we get capital T, small t. This side also capital T, small t. You can match this one, right? This side small t is there, small t is there, small t, small t, small t, small t. Here you understand here because these two boxes they contains capital T. So dominant trait is present. These two plants are tall, but this no capital T is present, means these two plants are dwarf. Here the percentage means phenotype percentage is 50-50%, 50-50%. We can say 50-50 means what? 50% phenotype is tall, 50% is dwarf. We can say 1:1, or we can say 1:1 ratio of capital T or small t means tall and dwarf ratio, right? And genotype also. Genotype means we understand there. That means what? It is 50-50%. 50% is small t, 50% allele of capital T, right? Same as to genotype. We can say 50-50%, 50% capital T and 50% small t is there. That in case of it is capital T, small t, we can write 100% is there, capital T is there, right? Now in third case, because we know here parents means small t, offspring is also small t, then what we get here? Only small t is present. That means all the squares they contains only small t. That means what happens here? Here 100% phenotype is dwarf. That means what? 100% small t is there. Offspring is of dwarf. So through test cross we understand which one is what is the ratio of genotypes. This one is all the plants are tall, phenotype and as well as genotype both are same. Capital 100% tall plants, 100% means only capital T is there. 50% tall, 50% dwarf. So 50% gamete, 50% means alleles are capital T, 50% alleles are small t, hybrid. Small t is there means 100%, phenotype is dwarf means 100% genes, alleles they are also 100% are dwarf. Small t is there, right? Now you can understand if suppose that I use here these parents offspring, these parents are small t. Recessive parent means small t here I use, right? Suppose that when we use dominant parent, then what we get? Then we get, look here, when we use a dominant parent, then we have no one, because in all the case we get only dominant, phenotypically dominant trait is present, right? Look here, then you understand easily. Here we know same as to this one, right? Upper side it is parent. Parent means now here I use parent, dominant parent, right? This side offspring is there. This is offspring, right? Then what we get? Capital T means phenotype. 100% phenotype is capital T. We cannot understand because phenotype 100% is tall, right? 100% phenotype is tall, right? And second case, if we use dominant trait, dominant parent, then we get here dominant parent is there, and this side capital T, small t, this is the offspring, hybrid offspring. What we get phenotype? It is capital T, because we have to match this T, right? This side T is there, T, this side T is there, this side small t is there, this side capital T, small t and capital. Here also we get, because capital T is present in all these square. Capital T is a dominant trait. That means what? Phenotypically, phenotype 100% phenotype is tall. So by seeing the phenotype means appearance, physical appearance, we cannot understand this one is hybrid or it also contains 100% phenotype tall. This one also contains 100% phenotype tall, right? Because both contains 100% phenotype tall, we cannot get any idea which one is hybrid or which one is tall. Okay, suppose that in third case, what happens here? When we use capital T means parent and small t, right? This side offspring, this side parents. What we get? Small t, capital T, small t, capital T, small t, capital T, small t, capital T. In this case also capital T is present in all the square. That means what? Here these are also 100% phenotype is tall. That means what? When we use dominant parent, then we get no idea about which one is hybrid, which one is a pure dwarf, or which one is pure tall, right? That's why we use only recessive parent, right? By using recessive parent, we get idea. Phenotype is equal to genotype, then we get idea which one is hybrid, which one is pure tall, or which one is pure dwarf, right? And that's why a test cross means a cross between offspring and recessive parent is called test cross, and you understand here we have no idea which one is hybrid or which one is pure dwarf or pure tall, but in test cross we get the idea which one is this one is 100% tall means 100% alleles are capital T. Here 50% tall means 50% alleles are capital T and 50% alleles are small t. Here 100% phenotype is dwarf means here 100% alleles are dwarf. Right? Do you understand test cross? Okay. One thing also there. Here, how Mendel is mathematically explained, which we read in this passage. In this passage, we need look here. When he crossed with, he simply explained, right? A gamete means if it is a pure breed, capital T, and small t, in a monohybrid cross, right? Capital T means only one type of gamete is formed, and it contains only capital T. And here also one type of gamete is formed, it contains only small t. In F1 generation, this one is F1 generation. In F1 generation, right, a hybrid plant is formed. Means it is because it is dominant trait, so all the plants are tall. Same as to parent, this capital dominant trait. This one is recessive trait, right? All the plants are tall. By selfing in monohybrid cross, here self-pollination, or we can say simply it is selfing or self-pollination. So, in self-fertilization we can say, right? Self-pollination. So, what happens here? In the next, that means in F2 generation, we have 4 types of plant. It is capital T, capital T, capital T, small t, capital T, small t, and small t, small t. Right? 4. Now you understand here, we have 4. Suppose that 4 seeds are formed. So probability, what is the possibility? In this case, we can say in 4, 1/4 each, 1/4. It is capital T, capital T. This, because 2 are there, we can say here 1/2, right? This, it is hybrid plant, capital T, small t, and this one 1/4, it is small t, small t. That is given. Here you can say how it is 1/2 I write here. Here I, we can write here 1/4, 1/4, right? Because both are same. So when we can say, I can say here when we add this one, right? So what we get here 1, 2. That he explained in the large passage, right? This one 1/4, 1/2, right? Means we can say 25%. This one becomes 50%, 50%, and this one becomes 25% possibility, right? And this is also a ratio. In mathematically, here you can understand here, because these are t plus t is equal to t square, right? Here what is here? 2 capital T small t, we can write here 2 multiplied by 2 things are there, right? And this side it is small t square, right? This one, when I add this one, no, is equal to 1, and that it becomes Hardy-Weinberg equation. Hardy-Weinberg equation. Also, it explains the Mendel's statistical or mathematical law. Binomial theorem. That he explained in the whole, right? Because here 2 types of gametes are formed. One contains capital T, another contains small t. And when they fuse with each other, So that means 50% chances that capital T can be fused with capital T only, or capital T it can be fused with small t. Same happens also here. Small t it can also 50% chances fuse with small t, or it fuse with capital T, right? This 50% chances capital T formation of capital T small t, and for this one 50% chances capital T small t or small t small t. That he explained in the whole process. Okay, do you understand? Okay, come to NCERT, right? Also in NCERT a figure is given, and through this figure they explain us, right? They explain us how a cross. Look, what question is there? Use Punnett square. Try to find out the nature of offspring of a test cross. What ratio did you get? So already I tell you here, right? You can see when we use this Punnett square in our right here, you can see this Punnett square. What ratio you get? Phenotype ratio is here 100% tall. Phenotype ratio of this one is 1:1 ratio, means equal ratio, 50-50% charge ratio is there. And here in case of small dwarf, 100% phenotype is dwarf. This is the ratio of phenotype, right? What ratio did you get? So for tall, for pure tall, we get 100% phenotype ratio. For hybrid tall, we get 50%. 50-50% means 1:1 or equal ratio between tallness and dwarfness. And when it is pure dwarf, then we get 100% phenotype ratio, right? Using the genotypes of this cross, can you give a general definition for test cross? Yes, we have a general definition. With general definition, when a offspring is crossed with recessive parent, then we get the genotype and phenotype identical ratio, similar ratio of genotype and phenotype, and that's why through this phenotype ratio we can get the idea about the genotype of offspring plant, right? That is here. In this figure you can understand homozygous recessive, and this side it is homozygous recessive, right? This one recessive, this one is also recessive, right? Okay, here what happens, this one is dominant phenotype. That dominant means here question mark is yes, and question mark refers that unknown. We have no idea. This one is capital T, means it is a tall, means it is a dominant character, trait, pure dominant trait, or it is a hybrid dominant trait. We have no idea, right? Because violet color is there, and violet color means what? Violet is the dominant over white color, right? So here we use WW, small WW, right? And for this one, capital W is given here. In this figure, on two sides, this is homozygous, this side. You can see here homozygous recessive. This one is one parent, and that is also homozygous recessive. This one is a parent. When a recessive parent is crossed with offspring, this one is offspring, unknown offspring. Unknown offspring. Then what happens? In first case, right? This side, these are gametes, small w from parent, and this one, if it is a here, it is given as heterozygous. Suppose that it is a heterozygous, right? Then what happens? One capital and one small is given. That is the genotype is this one. We have two types of gametes, one dominant and one is recessive gamete. Then what we get? Here capital W, capital W, capital W, capital W, small w, capital W, small w, because one capital letter is present. That's why they are violet in color, 50% violet, and this 50% these are white. Means we can say here 1:1 ratio between violet and white. 50-50%. That means what? Here genotype is also 50-50%. 50% capital W, 50% small w is there. But in this case, suppose that this one is dominant, that is dominant, pure dominant or pure breed line. Then what happens? We have a same type of gamete, capital W, capital W. This side is the parental gametes, or this gene is there, right? See what happens here. Capital W is present here, here also, this side also, this side also. In all the square, capital W is present, means a dominant trait is present. So all 100% are violet. That means what? When we see the genotype is also 100%. They contains 100% W, right? So through test cross, we understand that unknown flower is homozygous dominant, because all flowers are violet. All means 100% are violet. Here, half of the flowers are violet and half of the flowers are white. So unknown flower is heterozygous. You understand? You also understand how we get the idea. Okay, here it is not, white is not even, because white means only, we know white means what? White is a recessive trait and white is present only if in homozygous condition, right? So if white flower is there, we need not to test cross, because white comes only in homozygous condition, right? That means we need not to test cross with dwarf traits, right? Dwarf means recessive traits. Based on his observation on monohybrid process, Mendel proposed two general rules to consolidate his understandings of inheritance in monohybrid process, and today these rules are called as principle law of inheritance, right? This one is called principles or law of inheritance. The first law is law of dominance. Law of dominance means what? If we cross the two contrasting traits, two opposite traits or two complementary traits, right? Two different traits, then what happens in F1 generation? Only one trait is expressed, another one is not expressed. The trait which is expressed, it is called dominant trait, and the trait which is not expressed, it is called recessive trait. That law is called law of dominance, right? In F1 generation, it is proved by F1 generation. And the second law, that is law of segregation, or it is also called as law of purity of gamete. Law of purity of gamete, and it is proved by F2 generation, right? F2 generation, because in F2 generation what happens? These gametes, they get segregate, right? Without any blending, they get segregate. That's why it is called law of segregation. Without any blending, no blending is there. That means they are segregated in their pure form. So it is also called as law of purity of gametes. Okay. Now next point, law of dominance first. Characters are controlled by discrete units, and these discrete unit is called discrete unit. So characters are controlled by discrete unit, and these discrete units are called factor. Factors occurs in pair, capital T, small t. Single, they are not present, right? Third, in a dissimilar pair of factor, dissimilar capital T and small t, one member of the pair dominates, that is dominant, on the other, that other is called recessive, which is not expressed, recessive. So these three laws of dominance. First, characters, they are controlled by discrete unit, and this discrete unit is called factors according to Mendel, right? They are always present in pairs. And third is there, in a dissimilar pair of factors, one member of the pair dominates, right, or to the other or on the other, that is recessive. The law of dominance is used to explain the expression of only one of the parental character in the monohybrid cross in F1 generation. That is, law of dominance means F1 generation. Understand? And the expression of both in F2, it also explains the proportion of 3:1 dominant, that is phenotype generation ratio, obtained at the F2 generation, and that's why it explains the law of segregation, right? 3:1, 3 why 3 is there? The law, due to the law of dominance also, right? Because 2 in 3, in this 3, 1 is pure tall and 2 is their hybrid tall. Hybrid, they also explain. Hybrid means hybrid explain the law of dominance. Okay. Now law of segregation, second law of inheritance. This law is based on the fact that alleles do not show any blending and that both the characters are recovered as such in F2 generation, though one of these is not seen in F1 stage. F1 stage, law of dominance is there. F1 stage, both are present in the single zygote, right? So only one is expressed, another one is not expressed. But in F2 means what is given? Both the characters are recovered as such, right? Though the parents contain two alleles during gamete formation, the factor or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Only one of the two factors. Like in human male, only one of the two factors means one X goes in one gamete, another Y goes into the another gamete, right? Only one is going in, right? Of course, a homozygous parent produce all gametes that are similar, like in female, that is homozygous, XX is there, a similar type of gamete, or we can say single type of gamete is formed, right? While a heterozygous one produce two kinds of gamete, each having one allele with equal proportion, capital T small t, so means two gametes 50-50% they are divided, right? Now, incomplete dominance. When experiments on peas were repeated using their traits in other plants, it was found that sometime the F1 generation, sometime what happens, had a phenotype that did not resemble, this is not similar with any one of the parents, right? So that means what? They did not obey the Mendel's rule. They did not resemble either of the two parents as was in between two, right? The inheritance of flower color in a dog flower, it is also called as snapdragon, and scientific name is Antirhinum species is there, right? Is a good example to understand the incomplete dominance. In a cross between true breeding and red flower and true breeding white flower plant, small r, the F1 generation, that is capital R small r, was pink, right? And when the F1 was self-pollinated in F2, it resulted in the following ratio: 1:2:1. Here the genotype ratio were exactly as we would expect in Mendelian monohybrid cross. Look here. In case of incomplete dominance, we have same ratio, means genotype and phenotype both are same ratio, right? Here the genotype ratio were exactly as we accept in the Mendelian monohybrid cross. F2 in F2 generation. F1 generation means all the plants are pink. They are not resembled with any one of the parent, right? But in F2 generation, they segregate like 2, means they obey the Mendelian rule. 1:2:1. Means 1 it is red, 2 it is pink, and 1 it is white. But the phenotype ratio had changed from 3:1, right? Recessive ratio. What happened was the R was not completely dominant over R, that's why it is called incomplete dominance, and this made it possible to distinguish capital R, small r as pink from RR red and RR white, right? You understand this one? This ratio is given here, you understand. Red and white, F1 generation, all are pink. F1 generation, all are pink flower, incomplete dominance. This one is called incomplete dominance. F2 generation, this one is F2 generation, this one is F1 generation, right? In F2 generation, what happened? One is pure red, two they are pink red, right? Pink, and one is small t. You can see here phenotype ratio is 1:2:1, right? And genotype is also 1:2:1. It's 1:1, means both are similar. Both are similar, right? Okay. Next portion we will discuss in our next class.